15 of the singletons, and that means these are the ones that didn't cluster, had not been documented anywhere in any of the global databases. So then we sent them off to the WHO supranational laboratory at the Pasteur Institute. Even they couldn't figure out 4 of them. They couldn't assign international types. They couldn't assign them to any lineage that is known to date. Most of these actually were at least resistant to at least one drug. You know, one of the drugs that I use, that is rifampicin, isoniazid, ethambutol, and streptomycin, that we added also. And most of them were actually recovered from people who were HIV positive. 3.7% of the isolates were resistant to all the drugs that are in use today on the DOTS treatment plan. Most of them, all of them, were from the Uganda 1 subtype. Of course, there was also monoresistance to rifampicin and isoniazid and streptomycin, as well as ethambutol. The summary of this drug susceptibility test, we see obviously you would expect more resistance in people who have had prior bouts of TB. But then we found with ethambutol we have more resistance in people who are coming in as new cases of TB. They've never had TB before, and here they are showing up with strains that are already resistant to ethambutol. So there must be something sinister going on with ethambutol. Unfortunately, we didn't recover any non-tuberculous mycobacteria or M. bovis, and a bit more. The last paper documented polyclonal infections. This is a phenomenon where an individual, a TB infected individual, is infected with more than one TB strains. We found that 8 of the 72 that we analyzed actually had polyclonal infections, and 3 of these had compartmentalized polyclonal infections. That means they had one genotype in the pulmonary compartment and then another genotype in the extrapulmonary compartment. Take a case in point, this patient 8034. In the pulmonary compartment, this person is infected with the T1 family strain or isolate, and in the extrapulmonary compartment they're infected with X2 family. So obviously this individual, I mean the genotype in the pulmonary compartment cannot be the progenitor of what is in the extrapulmonary compartment, because these are they come from 2 different lineages. So this would simply mean this person, assuming this person was infected airborne, these 2 genotypes at one point in time must have been in the lungs and probably had a fight, and the XT2 family genotype decided to migrate to the extrapulmonary compartment. I'm putting it in a literal term, as this simply means there was a sequential infection process. It was exposed to one into the pulmonary compartment, exposed to another. These were something went on, and one decided to migrate to the extrapulmonary compartment. On the other hand, the other 2 patients were infected with Uganda 2 and Uganda 1 in different compartments. So chances are one could have been the parent of the other. It mutated into another, and then the mutant moved out. But that might not be the case for patient 668871, because the mutant or the genotype that he has in both compartments can be traced in the population that we sampled. So this could be the same case as patient 8034, that this person was sequentially exposed to these genotypes, and one of them actually migrated to the extrapulmonary compartment. On the other hand, patient 6697, this person has a genotype that is only exclusive to them in both compartments. That means, and this is from the same lineage, so the pulmonary genotype, it's possible that actually it's the progenitor of what is in the extrapulmonary compartment. And this microevolution is believed to be fostered by I mean, when infecting population of bacteria multiplies up to a certain level, then chances are, a spontaneous mutation can occur. So depending on how long this person gets treatment or gets diagnosed, this will determine how long this process can, and this is just one of the ways in which microevolution can occur, and might be able to explain why these individuals have more than one genotypes in different compartments of the body. The rest, they had more than one strains in the same compartment, that's the pulmonary compartment. But we found an association with gender. In epidemiology we use parsimony, use the data that you have. Obviously we had 72 samples with only 8 positives. This is already skewed. In order for us to fit a model, we need at least 40 positives to fit such a model, and of course 40 negatives as well. We have a way of analyzing such data. We use an exact logistic model, and this indicated that it's more likely that females are associated with polyclonal infections. And why is this so? I mean, in the typical African culture or setting, normally when somebody gets sick, it's the ladies who go to hospital to take care of these patients. So now imagine one of these 2 patients in the same ward has a different genotype of TB. These other three women who are having a conversation actually can be exposed to any of the genotypes these 2 patients have. So this could explain why there was an association of polyclonal infections with female. Now whatever we've been seeing is on individual basis. Now what would happen then in the population, on a population level? Now imagine these 6 patients. I mean, of course these 6 patients are going to give different microenvironments to the bacteria, and therefore different mutation pressures for the bacteria. Imagine this patient is infected with Uganda 1 and passes it on to the next, passes it on to the next, and passes it on to the next. We expect that if we take a sample right there, this Uganda 1 could have changed or mutated. But how we'll be able to account for what all these changes that have gone on along the transmission pathway? Conventional epidemiology, molecular epidemiology, actually has been documenting clustering, and clustering only indicates the strains that are circulating the most. But when we look within the cluster, it is possible to see what the changes that are going on within this cluster, and we believe this would explain microevolution at the population level. Now if we assume that the 344 patients that we sampled are representative of the TB prevalence in the population, that means we're able to see how the genotypes that are circulating the most are evolving within the population. And that we see that the analysis we did, we see that Uganda 2 is evolving, is much more homogeneous. It's not evolving as fast as Uganda 1, and yet Uganda 2 is the most prevalent. We used minimum spanning tree. The green shows samples that were recovered from the rural areas, and the purple represents samples that were recovered from the urban areas. The thickness of the line connecting any two of those structures represents how close the structure is. If the line is thick, then that means these two are closer to each other. I mean, we use the difference of a single locus variation, so they are closer to each other at a single locus variation basis. Now, having that in at the back of your mind, you see that actually Uganda 1 has more thicker lines. That would mean Uganda 1 is much more homogeneous. It's this isolates within Uganda 1 are closer to each other, which is the opposite for Uganda 1. Of course, you see thin lines in Uganda 1 and just few, or actually 2 thick lines. Conventional knowledge would simply dictate the opposite. Since Uganda 2 is the one that is more prevalent, we expect it to be changing more. But if we critically then look at Uganda 1, you see that majority of the Uganda 1 isolates were recovered from individuals from the rural areas. That would simply mean that either there are more exogenous additions of Uganda 1 to the rural areas. And under what circumstances would that happen? Most of the Ugandans who live in rural areas actually hail from rural areas, and when they get debilitating diseases like HIV and AIDS, towards the terminal stages of the disease, they are actually transferred back to their rural areas. So these might be, this exodus might be the source of exogenous strains of mycobacterium of the Uganda 1 genotype, which is much more prevalent in Kampala. Also, the dogma that fosters microevolution dictates that the longer you take to diagnose TB, the more time you give the infecting population to evolve and mutate. And this is more likely to occur in rural areas. I mean, rural areas, they are pretty far from the hospitals where this diagnosis can be made, and therefore for them to start on treatment. So it is understandable that the microevolution would be expected to happen more. Microevolution due to the lag of diagnosis would happen more in the rural areas, and that would explain probably why we have more of these Uganda 1 heterogeneous genotypes from the rural areas. And we also know that 65% of rural Ugandans actually visit the traditional medicine and before they actually go for the allopathic medicine. So all these social factors seem to be also playing a role in the molecular picture that we see in Mubende district. And this would definitely mean that in order for us to combat tuberculosis, we cannot only look at the medical part. We have to look at the social anthropological aspects of the disease, put every facet together in context. So the conclusions of this study shows that water and soil harbored most NTMs, and that animals had a key role in contaminating the environment. Precipitation is known to play a key role in adjusting the load in the environment, but valley dams were deemed to be the most risky sources of water, and therefore if humans are drinking untreated water from these areas, chances are they're going to be exposed to non-tuberculous mycobacteria. The animal study shows that pigs are terrestrial filter feeders accumulating mycobacteria as they scavenge for food, and pigs too are more likely to acquire it from these valley dams or from animals. Routine pork inspection has a low sensitivity, therefore there's quite a lot that will be missed. So if and when humans consume undercooked pork, chances are they're going to be exposed to non-tuberculous mycobacteria, but also tuberculous mycobacteria, because we isolated bovis in there. The molecular characteristics of some of the strains seem to be just influenced by host factors, environmental factors, or geographically enhanced factors, and therefore for anybody to develop a geographically tailor-made diagnostic tool, they will need to put this in consideration, not to simply pick a tool that has been developed somewhere else and simply think it will work just as well as it has been working where it was developed. The case study in humans, we found the high prevalence of TB in HIV-infected individuals who are smoking, but also living in households that are much more crowded. MDR, TB, and high medical-related transport bills will be, and are most likely to be, the bottleneck to the current control strategy. And given that we isolated a high number of genotypes, including those that are known to be restricted to certain geographical areas, is an indication of dynamic but also complex epidemiology of TB in Mubende district. Polyclonal infections are an integral part of these dynamics. The recommendation then will be avail safe water to these pastoralists to prevent them from